here, and make sure this goes into. So my name is Amy Beaulieu. I'm on faculty here at NJMS. I am an immunologist by training, but have a pretty diverse history in both immunological host defenses as well as microbial pathogenesis. And I did my PhD actually on Mycobacterium tuberculosis and understanding virulence factors produced by the bacterium that causes TB, and how those virulence factors affect the host immune response. So for today's lecture, we're going to keep it actually pretty broad and foundational so that you understand some of the basic concepts underlying pathogenic mechanisms and pathogenesis in general. At the end of the lecture, you are always welcome to reach out to me if you have any questions or would like to meet for office hours. I'm located in the Cancer Center on the NJMS campus, room G1222, or you can reach me by email at this email address here. And I will note that I've made just a few minor updates right before walking in this morning, so including addition of my name and contact information. So I'll upload these revised slides right after the lecture as well, so these little details, you have those at the end. But by and large, the lecture slides you have from this morning have the information that's needed for the class. So today's class, in terms of what we'll be covering, we'll cover 4 major topics of pathogenesis with a focus on bacterial pathogenesis. We'll talk about establishment of infection in the host, how the bacterium interacts with host defenses to avoid those defenses and successfully establish infection. We'll talk about classes of molecules known as toxins that are produced by bacteria and often have really remarkable effects on disease progress and outcome in the host. In some cases, those toxins are also virulence factors that help the bacterium survive in otherwise hostile niches. And then at the end of the lecture, we'll spend just a little bit of time talking about 3 organisms, very well-known organisms, and some of the diverse roles that they play in causing disease, as well as some of the very specific mechanisms of pathogenesis that they employ to be successful in their disease on the host. So concurrent with the lecture outline, at the end you should be able to describe in a general sense how most infections are established, describe several important mechanisms by which microbes avoid the host system, the host immune system, and the host defense system. This should be 1, 2, 3, 4, by the way. I'm not sure what happened here. Describe the mechanisms of the main classes of bacterial exotoxins, and I'll tell you a little bit about what the difference between an endotoxin and an exotoxin is. And describe the etiology of disease, the diagnosis, the mechanism of virulence and disease-causing activities, as well as treatment courses for several very important clinically important bacteria that are known to cause disease in humans. And I expect that most of you will have heard of these organisms and know at least in a general sense about these diseases that I'll describe. Okay, so really broadly, how does a microbe establish infection in the host? This might sound really simple. Actually, it looks like the numbering system is messed up for a lot of these slides, so just assume it's one through whatever on the slides. I'm not sure. There must be a difference in the format here. But just in a general sense, how does a microbe establish infection? And these really, I think, are very obvious but key points of the progression of establishing an infection. The first thing that the microbe needs to do is, of course, encounter the host. Once it has encountered the host, it usually undergoes a series of steps that involve both adherence to the host surface. Often a pathogen, you'll learn, is entering through surfaces that are lined with a border of epithelial cells that the pathogen will need to interface with and either adhere and colonize on the surface of the epithelium boundary or actually subvert that boundary and enter the underlying tissues or bloodstream where then it can go on to multiply and colonize the host. So once it has either adhered and or entered the host tissues, it will undergo a process of multiplication and colonization. And so multiplication can happen really anywhere, whether it's the site of colonization or a distal site first. Colonization usually occurs at the landing site of the organism, the part of the body where it will stay and expand in numbers rigorously. And then from there, occasionally some microbes will additionally spread to other distal sites in the body. And in some cases, this is really the most dangerous part of the infection, especially if the microbe gains access to the blood system or the bloodstream, where it then can disseminate across the organ systems, and in that case, usually leading to very rapid death of the host. So septic shock is an outcome of bacteria or bacterial toxins that have gained access to the bloodstream and now are disseminating across the body systems, where immune responses often are too vigorous for the organs to sustain vitality. So in addition to spreading, both local and disseminated sites of infection will cause damage to the host tissues. And again, this is usually the process that causes the symptoms of the disease that you'll encounter for most of these microbes. And then the final, the end point of all of this really is much like that of a battle. Either the microbe wins and the host dies, the host wins and the microbe is eliminated or at least kept in check, or they enter a state of equilibrium where they will coexist without any notable disease arising in the host. So sometimes this is entirely non-clinical symptoms, or very low-level subclinical symptoms that the host can maintain a normal, or at least somewhat normal lifestyle from there on. So types of encounters with most microbes fall into one or two categories, those that are exogenous and those that are endogenous. And as implied by the names, the exogenous encounters are typically occur when the host interacts with microbes in the environment. These might be microbes or bacteria that are found in food, water, soil, or transmitted as a result of insect bites. These are also many microbes that are transmitted between hosts, especially human hosts, as a result of dissemination of body fluids. So this might occur during sneezing or coughing if you have a cold. These can be sex-delivered microbes that occur in the context of sexual encounters, as well as fecal-oral route of transmission, and which is typically one in which the microbes are transferred from fecal samples in the environment, encountered in all kinds of places, to the hands, and then the hands, in the context of eating or drinking, transfer it to the mouth of the new host. In addition to these many environmental encounters, all of us in this room have a whole host of different microbes that live on and in us all the time. And in most cases, those microbes fail to cause any disease whatsoever, partly because some of our host immune systems are very good at keeping them localized in places where they are harmless or unable to grow to a point where they could cause disease. And in other cases, these might be microbes that actually have, under the right circumstances, a positive role for the host. And so this is true for many of the microbiota in your gut, which play very important roles in helping you digest food and other scenarios. Although, in some cases, if they get to the wrong place or they become too florid, if there are too many of them, that they disrupt the equilibrium in the tissues, they actually can cause disease in those cases. And so it's important to keep in mind, of course, as adult or mobile individuals, we're constantly moving around our environment, encountering pathogens, many different classes of pathogens throughout the course of our day. But really, your exposures start almost immediately at the onset of life. And so there are some microbes that actually you encounter in utero during placental development and fetal development. Most of those in a healthy pregnancy are not disease-causing for either the mother or the fetus, though in some unhealthy pregnancies, that's not always the case. And then even more so during the birthing process itself, there are a number of microbes that actually are resident in the vaginal canal that are then transferred to the baby as it's being delivered. And again, some of those microbes are good. The birthing process is important for seeding a lot of the gastrointestinal flora that will then become part of your natural microbiota and help the baby in processes of food digestion, as well as overall health of the intestinal tissue. But some are bad. Some are potentially disease-causing. And the baby is often protected against those disease-causing organisms by maternal antibodies that are transferred to the fetus during fetal growth growth and then exist in the baby after birth, and provide protection in the early years, particularly the early months, when the baby usually has a very under-functioning immune system. It just hasn't matured to the point where it can defend itself. Now drilling down into the distinct steps of gaining entry to the host and progression of disease. There really are two major ways that an organism gains entry to the host, and they fall into the categories of either ingress or penetration. And I'll talk to you a little bit about what each of those distinct pathways look like, but they basically get down to the bottom line of whether the organism is going to grow on the epithelial surfaces that protect and provide boundaries of the body, or whether it's going to transverse those epithelial surfaces and gain access to the underlying tissues or even the bloodstream. And so penetration usually is a process where the organism can transverse or go across, move through the epithelial boundaries and borders that line all of your organs and tissues. Whereas ingress, the organism will typically grow on top of those surfaces and still cause disease, but from on top of the epithelial border. And this really boils down to the way that the human body and mammalian body is organized, where you have both the outer epithelial border that is your skin that protects, that provides really the harsh, dry outer shell of your body, as well as many inlets and outlets, and these are your mucosal surfaces, that are both contiguous with the interior of your body and the outside world. And these include, so for example, your mouth and your nose connect to your lungs. The urogenital tract will connect into the uterine compartment and the kidneys. You have the eye socket, which is really a wet mucosal surface where internal in the socket there's actually access to the internal compartments of the host as well. So all of those areas are places where an organism doesn't necessarily have to cross the border in order to gain access to the interior of the body. And so mucosal organs we'll talk a lot about, and mucosal organs that many organisms use to gain entry include the intestinal compartment. And this is the favored route of entry for organisms that are carried in food or in water. The lungs, these are microbes that often are transmitted by sneezing or coughing among individuals in transmission. And then, of course, the urogenital tract. And many sexually transmitted diseases actually first encounter and gain entry to the host via this route. So ingress. As I mentioned, ingress is really a fancy word for saying that the microbe gains access to the human body as a host, but it doesn't necessarily cross the epithelial barriers that line the mucosal organs or the skin. And there are actually a large number of pathogenic molecules that are perfectly happy growing on the exterior surface of the epithelial barriers, including in the lumen of the gut, the lumen of the lung, or the lumen of the urogenital tract. So it's not actually crossing those barriers so much as colonizing the surface and doing damage from there. So we'll talk about two right now in particular, two that are really relevant to later parts of this lecture and other parts of this course. The first being your lungs, where many microbes gain entry via inhalation. And this could be inhalation of aerosolized droplets when another individual who's already carrying the microbe sneezes or coughs in your vicinity. You breathe in, and now those microbes gain access to your lungs. In addition, dust in your environment actually carries a number of different pathogenic microbes that can gain entry when you're just normally breathing. So it doesn't even have to be close to another sick individual. You can gain access to certain pathogens this way as well. And so not surprisingly, as a favored route of entry of many different microbes, and the fact that you can't really do anything to stop breathing. You have to breathe. And so by virtue of having to breathe, microbes will automatically be coming and going very frequently and rapidly. So the host has devised a number of different mechanisms to protect against infection in this compartment. And these include the ciliated epithelium. So these are the cells that form the border of the lung inside of your lung compartment. And the ciliated aspect refers to little hair-like structures that actually grow out into the luminal side, so the top surface of the lung compartment, and you can see them here in this diagram. And by constant motion, and this is a very active process, they are constantly and continually sweeping fluids and microbes and particulate matter up and out of the lung, usually into the upper GI tract, so that those particles, mucus organisms, can be swallowed and destroyed via that route. So the cilia are very, very important in basically constantly sweeping out this compartment and keeping organisms out of there. In addition, on top of the cilia, there's often a very thick and sticky mucus layer that has different surfactants produced by various immune cells and non-immune cells in the lung. And so this mucus and these surfactants, which kind of keeps a slick, slippery surface in the lung, are able to grasp the microbes that might gain entry, and as they're being swept out by the cilia, it's basically a big gooey mess that's carried away, and it keeps the surface of the epithelium relatively clean and impervious to the microbes that might enter via this route. So in addition to the mucus and these surfactants that keep things moving up and out of the lungs, you have a number of antimicrobial peptides, proteins, and other factors that are actively secreted into the fluids in this compartment. And these are often broadly lytic for many different classes of microbes, so that almost any, not almost, but many, many different types of microbes that enter in here are immediately lysed and killed, even before having a chance to land on the top of the epithelium surface. And then there are many different types of immune cells that patrol the lung compartment. A few of them actually crawl out into the alveolar spaces, so they're actually on the luminal side, where the air will first come in, the microbes will first come in. And many of these immune cells are very adept at sensing, engulfing, and killing microbes that they encounter the environment before those microbes have a chance to replicate. And so this host system is really extremely efficient and well orchestrated to protect this entry point. Similar defenses exist in the GI tract, which is another very common route of pathogen acquisition by virtue of the fact that we must eat and drink to sustain life. And of course, many of the substances that we eat and drink are very nutrient rich, because that's why we're consuming them. And those nutrient rich sources of food also will support many different microbes, the life needs of many different microbes. And so you have a lot of bloodborne and waterborne bacteria that are constantly encountering your GI tract due to the fact that they're in your foods and drinks. So similar to the lung, there are many host defenses that the body has set up to protect against these organisms. These include the most obvious being stomach acid, which is an incredibly acidic, harsh environment that few organisms can get past to gain access to the lower intestinal system, which is a little bit less harsh. There are, in addition to the acids produced in the stomach, there are the digestive enzymes and the bile salts. And so all of these create a very harsh environment that few microbes can survive. Those that do gain deeper entry into the GI tract and enter the intestinal system are then swept out through a process known as peristalsis, which is a wave-like contraction of the intestines that actually are important for pushing food through your digestive system, both prior to nutrient acquisition from it, and then of course through the waste production that happens at the end of the GI process. But this peristalsis is a constant movement that also helps carry microbes out, much like the cilia, the ciliated epithelium in the lung. It's constant movement that really prevents bacteria, many bacteria, from gaining access and sticking for very long periods of time. That peristalsis, much like in the lung, is often coated with the surface of the intestinal epithelia is coated with a very thick layer of mucus as well. So it protects access to the epithelial cells that form the border of the intestinal tract, and it actually captures and encapsulates most microbes so that those can be swept out and excreted in the downstream of the peristalsis movements. And so then the last thing to keep in mind is, much like the lung, there are many different immune cells as well as non-immune cells that are embedded in the epithelium border that constitutes the GI tract surface, as well as right immediately underneath that epithelial border in your intestinal system. And these immune and non-immune cells secrete all kinds of antibacterial, antimicrobial factors, some of which can directly lyse pathogens that enter in that tract. So again, a very, very active host defense system is engaged in the GI tract. So those are two very popular methods of ingress for microbes that cause disease. And then, of course, there are a whole host of other organisms that gain entry through penetration of the epithelial borders that either line your mucosa or, in fact, are your skin on the outside. And so two main classes of microbes that gain entrance via penetration include microbes that are injected by insects. So malaria, for example, is a microbe that can't necessarily enter your body system until it's injected by its carrier host, the mosquito. Breaks in the skin also allow different microbes that normally could not traverse the epithelial barrier or boundary to gain entry to the subepithelial tissues where it might be more amenable to growth of the bacterium and dissemination into deeper sites into the host. Because of modern medicine, we actually are often transplanting and trading fluids and organs that normally would not gain access to another host. And by virtue of blood transplants or organ transplants, a number of different pathogens can be transferred in that process. These include HIV. Many of the hepatitis viruses can be carried through bloodborne transfusions, as well as a number of other organisms. Sorry about that. So those are our popular entry methods. And then, of course, viruses. So many classes of viruses have developed machinery that allow them to bind to receptors. So here's a viral particle in this diagram here. And this is really a general schematic of how a virus gains entry to a host cell. So viruses have proteins on their surface that will directly engage receptors encoded by host cells. And this can be the epithelial cells that that comprise your skin or line your mucosal organs. And by virtue of engaging those receptors, the viral particle is actually taken up by the host cell. It transverses the host cell membrane and enters the cytoplasm of the host cell, where it then can be replicated. Generally, this is now a replication event for the virus. Additional copies of the virus will be made by the host machinery. It will be encapsulated into a viral particle sheath, essentially, and then it is extruded by that host cell back into the environment through a budding process that then gives release to another mature viral particle. And so you can imagine this process where the virus gains entry to a cell type that is at the border of a mucosal organ, the epithelial cells, or your skin. When it's extruded on the other side, it doesn't necessarily have to go back into the side of the body that is the exterior, but rather can be extruded into the interior. And so gain access to the tissues that are underneath those epithelial borders and the interior of your body. So viruses are very, very adept at crossing any kind of border that we try to use to keep them out in the system. Okay, so in addition to this process of entry, a lot of organisms or microbes are not actually able to establish a site of infection and undergo colonization, or even able to penetrate the epithelial barriers that line your organs without first undergoing a step of adherence. And this is particularly important in your organs that have mucosal layers that really form this really thick, sticky outer network on the exterior surface of the epithelium that prevent the pathogen from gaining access to the cells that are underneath. So in order for a microbe to gain access to that cell, stick and replicate, or stick and penetrate, it needs to adhere first. And this is actually one of the most rate-limiting steps in a pathogenic organism causing infection. And so to get around this process of the host constantly trying to sweep them out and entrap them in sticky fluids, many bacterial species have developed proteins known as pili which are little hair-like, protein rod-like structures that stick out of the surface of the bacterial, usually the outer membrane of the bacterial wall. And these pili are very sticky for different receptors or classes of molecules that might be expressed on the surface of the host cell. And so by extruding many of these different pili, engaging many different receptors on, or many copies of receptors on the host cell exterior, it can actually begin to stick and then firmly adhere to the surface of these epithelial or non-epithelial cells that it encounters at the borders. And so pili are one major class of adherence proteins we'll talk about. There are other non-pili shaped adherence proteins. So here's an example of what those might look like, where instead of this rod hair-like extrusion being the protein produced by the bacterium that sticks, it's actually more globular domain proteins. But nevertheless, the idea is the same. These globular proteins will stick to other proteins or other molecules on the surface of the host cell. And by virtue of many different interactions allowing the sticking to occur, you can actually get a pretty tight adherence to the surface of the host cell. And so these tethering molecules, as I mentioned, are often proteins. They will bind to target receptors or even sugars or lipids on the host cell surface. And all of this process is really about preventing the host fluids from washing them away. This includes the mucus or for example, in your eye sockets, tears. But all of this helps prevent your host fluids from washing out the microbe. And for example, Neisseria gonorrhoeae, which is the organism that causes gonorrhea, sexually transmitted disease in humans, has a very particular pili that will bind to the urogenital epithelial cells and allow it to stick, colonize, and really establish a proper infection in that compartment. So if an organism has managed to successfully enter the tissues, adhere to the tissues, its next step is really to multiply as quickly and adeptly as it can, to colonize that surface if the site where it's landed is actually its preferred site, or to disseminate to another site in the body where it prefers to grow. And then in some organisms will then further spread throughout the body and cause systemic disease. And so all of these factors are very organism dependent. They depend on the nutritional requirements of the pathogen relative to the site where it actually gained entry, the pH of that compartment, the temperature of that compartment. For example, your skin is a much lower temperature than your internal organs, and it may or may not be the optimal temperature for a bacterial to grow and replicate. And then depending on the nutritional and metabolic requirements of the pathogen, some can survive in many different niches in the host, and others require very, very specific tissues that foster their nutritional needs. In addition to this process of multiplying, colonizing, spreading, many bacteria have developed the ability to form biofilms. So biofilms are very interesting. It's a partially living film that will often coat the surface of the colonized tissue or organ. And it is comprised of the pathogen itself, as well as polymers of fibrin in the environment, and usually many sticky sugar molecules, polysaccharide molecules. And it creates a very sticky net of bacteria that do a few things for the bacteria. Often the sticky net will prevent immune cells from effectively getting into the core of the net to kill off the microbes. The sticky net will actually prevent a lot of these secreted molecules of the host system from gaining proper entry to the bacterium or underlying tissues. And something about the biofilm process often changes the metabolic activity of the bacteria as well, so that they are rendered less susceptible to a lot of the antibiotic treatments that we use in the clinic that actually target metabolic enzymes of bacteria. So it's altering their metabolism in a way that they're actually less susceptible to the antibiotics that we use clinically. So this is a very effective adaptation strategy. A number of different bacteria use it. These include the bacteria that will eventually cause what you experience as dental plaque. And dental plaque begins as a biofilm that over time will harden, but it's actually it's caused by a bacteria that's colonizing your gums and the surface of your teeth. A number of other bacteria will often, for catheters that are inserted into patients for long periods of time, they'll form this sticky net over the interior of the catheter, and that's just a site of continual seeding of infection for the patient as well. And then different plastics that we use in surgical procedures. So these might be joint replacements or heart replacements or heart valves, so things like that in the body. If those pieces of foreign material at any point during the implantation process get a bacterium on them, for some reason bacteria love plastic surfaces, and they'll often form these biofilms over these implanted pieces, implanted surgical devices like heart valves. And often the only way to undo a biofilm on a heart valve is to cut it back out and put in a new one, because they're really difficult to clear with just traditional antibiotics once that biofilm forms. And so here's an example of a biofilm on the interior of the aorta lumen. And you can see if you really look at these closely, you can see the bacterial rods here, and then it's just this sticky net of polysaccharides and host proteins that have been trapped. So it's a pretty effective strategy overall. All right, so it's gained entry. This pathogen has gained entry. It's adhered to the surface or penetrated into deeper tissues. It's multiplied. It's spread. And so now you have the part that is experienced as disease. So this is the part where you typically begin to experience clinical symptoms. Those can be mild symptoms like a cough or sneezing, or very severe symptoms such as limb paralysis, depending on the organism that has gained entry. So depending on the organism, whether it's a bacterium or a virus, or for example, a eukaryotic pathogen like a parasite, you can have local and systemic tissue damage. So for example, there are a lot of helminth or worm pathogens that will gain entry to the host by burrowing through the skin layer. They then will traverse to the lung, undergo a part of their cycle where they grow and mature. They'll then break through the lung tissue and just eat holes out of it, gain entry to the gastrointestinal system, where they then will replicate, undergo sexual reproduction. You'll get more tissue damage in the GI tract. They're excreted, and the process starts all over again. All of those steps are really, from a host disease standpoint, you have serious tissue destruction in the skin, serious tissue destruction in the lung, and then in the GI tract as well. And then that's because the worms are really literally burrowing through these tissues and destroying it as they go about their business. So you can have mechanical destruction this way. You can have cell death. So a lot of viruses will infect a cell, replicate, and eventually the cell, because it's overwhelmed by the production of all of these foreign particles and proteins, will simply die. You can have bacteria that will actually inject toxins into host cells and kill them that way. You have, as I mentioned, a number of different toxic molecules produced especially by bacteria. These might help the bacteria spread, colonize. They might stop cell growth. In some cases, the bacteria will deliberately lyse the cell in order to recover nutrients that are housed within the cell. For example, organisms that require iron reserves, which is usually in relatively low concentrations in the body, need to release those iron reserves from cells themselves in order to get access to them for the bacterium to keep growing. And then, of course, there are a lot of different toxins that will act on host cells and exaggerate their normal processes. So, for example, the botulinum toxin and the tetanus toxin, which are both produced by Clostridium species bacteria, work on nerve cells and actually cause, they overexert the nerve cells, so that, so with botulinum, you get flaccid paralysis, so the muscles stop tensing, whereas in the tetanus toxin will actually cause muscle tension that is so severe that the lungs can stop working, or the spine can basically turn in this very crooked orientation, or the muscles that hold the jaws, that allow the jaws to work, become so tense the individual can't open their mouth to eat or drink. And so these are all the outcomes of different toxins the bacteria is producing that are working on specific host cells, either to exaggerate or shut off specific cellular processes. And this can't be underestimated, your host immune system is geared, it's completely programmed to identify pathogens, target them, and kill them. And a great majority of that process is driven by inflammation. So inflammation from an immunological standpoint is secretion of proteins that cause inflammatory outcomes in tissue. So this could be swelling, so fluid retention, which manifests as swelling. This could be influx of new blood cells, which manifests as redness in tissue. This could be pus production, which is important for killing extracellular bacteria. So all of these processes that you experience as disease symptoms are often caused by the host producing factors that are targeting the bacterium or the other pathogen, but end up causing all kinds of ancillary bystander damage in the host tissue itself. And this is a very prominent cause of a lot of the disease symptoms and tissue damage that arise as a result of infection. It's not actually the microbe itself, it's your host immune system trying to kill the microbe that's causing all the damage. So the two go hand in hand, and disease is the outcome of either or, really. And so that really leads to what can be considered the end game in this whole process. Either the microbe wins, and in some cases this is death for the host. The host can either eliminate the microbe or keep it in a state of check so that the disease itself, the disease symptoms are cleared. So some organisms are completely cleared, like for example, a strep infection in your throat, you might completely clear. But other organisms aren't completely cleared. They're just beaten back into niches where they're not causing clinical damage. So often an individual, if you're colonized with Staph aureus, which can cause skin infections, most of us actually carry Staph aureus everywhere on our body. But it's kept in such a way that it's not causing any sort of disease in the absence of any disruption to your tissue barrier. Mycobacterium tuberculosis is a bacterium that invades the lung tissue and causes TB, which, as many of you know, for centuries and centuries was a lead killer of humans because we didn't have effective antibiotics to treat it. But in fact, of the, let's say, 10 hosts that TB will infect, it actually only causes disease in about one of them. So only 10% of infected individuals will have disease. The others will typically keep the bacterium in a latent state, a subclinical latent state, where they're not experiencing any overt disease symptoms at all. And this can be even for their lifetime. They can carry it in this state or for long periods of time, decades. And then certain environmental triggers might trigger the exit from latency, and then active disease develops. So this is an example of the host winning but not clearing, and is possibly an example of coexistence, although in many cases those pathogens aren't doing anything good for the host, they're just waiting. There's also a coexistence, as I mentioned, with your normal microbiota, where the bacteria in the right numbers and in the right location might actually have very important functions, like metabolism by the microbiota in your GI tract as well. So these are all outcomes that the host is continually grappling with. And it's important to keep in mind that you're constantly inundated with pathogens in your environment, in all of these organs and tissues. And clearly, we're not constantly in a state of illness. So most of the host defenses are pretty effective. Okay, so I've mentioned the host is really great at evading microbes, or at least keeping them in check. Well, clearly the microbes that cause disease have figured out very clever ways to avoid these defenses, and they are numerous. And so the more successful a pathogen is at avoiding host defenses, the more likely it is to be a disease-causing microbe. And so some of the ways that microbes evade host defense, and there are so many ways, but we'll talk about 5 different kind of common classes of avoidance that a lot of different pathogens will utilize to avoid being cleared by the host defense system, which is typically mediated by host immune cells, but non-immune cells can contribute as well. So these include evading a process known as phagocytosis, and I'll take you through what that process is, but it is an immune process mediated by immune cells. inhibition of the phagolysosomal fusion, and escape from the phagolysosome to engage in intracellular replication. And so all three of these are really about evading different stages of the phagocytic process, which is mediated by immune cells such as macrophages, for those of you who have had any immunological training. So macrophages are immune cells that mediate phagocytosis, as are neutrophils. Those are two really key cell types that do this. In addition, especially at mucosal surfaces, you have secretion of an antibody known as IgA. And this is a very special type of antibody. It's typically found in very, very low concentrations in the blood or in the organs, but in the mucosal surfaces, and especially in the mucus, it is highly abundant. And it's particularly good at coating bacteria or microbes. And that coating process helps them be eliminated by the host immune system or by other processes in the body. And then the last one we'll talk about today is this process known as antigenic variation. So your immune system recognizes molecules that are conserved either in a specific molecule on the pathogen or by many molecules that are pathogen associated. And so by changing those molecules, often they're surface molecules on the bacterium that the host cells can see. So by changing those molecules, either changing their structure or whether they're on the surface, they can actually evade host detection or surveillance in this. And that process is known as antigenic variation. And I'll give you some examples of this as we go through. Okay, so what is phagocytosis? It's a fancy word that effectively describes the process by which an immune cell, and I mentioned there are a few that do this well, macrophages and neutrophils, by which the immune cell literally engulfs, eats, and digests the pathogen. It is truly a process of consuming and breaking down. So the process usually starts by the host cell, and you can imagine this as a macrophage or neutrophil, which do this very well. It will encounter the microbe, often a bacteria, in the environment. And by detecting the bacterium, often with cell surface receptors on the phagocyte, it will enclose it with its membrane on all sides. And so the microbe is effectively encapsulated in this vacuole inside the phagocyte. So you can imagine this white space in here is actually the extracellular fluid that got trapped in as the membrane of the phagocyte engulfed it, encircled it, and closed back in on itself. So you have an internal vacuole now. So all by itself, this internal vacuole is a pretty mild environment. It's mostly comprised of the nutrients and the pH that were in that extracellular fluid to begin with. So usually if the bacterium were floating around in extracellular fluid, it wasn't being killed particularly well by it. So it's evading that. But professional phagocytes, such as macrophages and neutrophils, have a hidden arsenal in the inside of their cellular compartment. And most of that arsenal is actually withheld in these intracellular compartments known as lysosomes. So lysosomes are specialized organelles, and they contain a number of different factors, enzymes, molecules that are very good at and quite dedicated to killing things in other compartments in the cell. And so the way that the contents of these lysosomal organelles gain access to the phagosome is a process known as phagolysosomal fusion, where literally this organelle that is the lysosome will move next to and merge membranes with this phagosome that contains the engulfed microbe. And in that fusion process, you get a new organelle known as the phagolysosome. And so the phagolysosome, unlike the phagosome, is really much more like the harsh interior environment of the lysosome. And so what do I mean by a harsh environment? The contents of the lysosome, as well as the recruitment of different factors to the phagolysosomal membrane, will cause a dramatic drop in pH in the interior of the lysosome, the phagolysosome, and it becomes an extremely acidic environment. You also have, by virtue of the lysosome fusion, and again the recruitment to the membrane of this phagolysosome compartment now, the production of reactive oxygen and nitrogen species, and these are literally just free radical oxygen and nitrogen species that will bind to anything, including microbe molecules, and inactivate them. And in addition, most lysosomes, and this really is a little bit cell type specific, will contain a whole arsenal of specific antimicrobial peptides and proteins, many of which will insert in the bacterial membrane and lyse the bacterium itself. And so all of this process leads to the outcome for the engulfed microbe that it is effectively digested. And then those digested bits can either be used as fuel for the phagocyte or can be extruded as antigens into the environment that no longer have any harm potential to the host. So this is a very important process and is extremely active. So there are many, many phagocytes that line your mucosal organs. These cells are abundant in the lung, in the urogenital tract, in the GI tract, and right underneath the surface of your skin. So these immune cells are in active patrol so that they can orchestrate exactly this function. And so there are lots of ways that the host immune system interfaces with phagocytes and enhances their function. And so just a broad idea of how the immune system works, you can kind of think of it as two different camps or classes of immune cells. Those that are part of the innate immune system, and these actually do include the phagocytes I mentioned. Macrophages and neutrophils are innate immune cells. And then you also have an adaptive immune compartment. And really the key players of the adaptive immune compartment are B cells and T cells. So imagine most of you know that B cells are the cell type in the body that produces antibodies. And so they're a very important target of vaccination. You want antibodies produced by B cells that then neutralize bacteria and prevent infection, or neutralize their toxins and prevent disease. And T cells play a number of roles we won't cover in this class, but they're considered part of this adaptive immune component. And so the important distinction from a pathogenesis and infection standpoint is that innate immune cells act immediately. Really, they're right there at the surface. They're poised for action. They're making effector molecules. Often they keep them inside the cell, but they're making them so that they're ready to release them as soon as the infection or the microbe is sensed. Whereas adaptive immune cells are held in a state of check, and they're completely inert in many respects early on. And they really require a lot of different signals that happen over the course of days or weeks to become activated. So this is a very slow process. And few adaptive immune cells, unless you've been immunized, are prepared to combat infection immediately. So one of the things that they do that adaptive immune cells do is B cells produce antibodies. And innate immune cells produce a bunch of factors many of which are secreted, although some of which are held on their membrane. And those factors sense microbes or bind to microbes. And antibodies actually do the same thing, but they're produced later in the infection, and they will bind to the microbe. And the process of these immune-derived factors, and most are proteins, immune-derived proteins, including antibodies, binding to the surface of the microbe, is that they coat the microbe with these host-derived factors, and then those host-derived factors are very, very readily recognized by receptors on the phagocytes. So the neutrophils and the macrophages have receptors that look for microbes that are coated with these other host factors. They sense them, and then that triggers the process of engulfment and phagocytosis and ultimately killing of the microbe. So this is probably your 2-second overview of many, many, many different processes in the host immune system interfacing. But just keep in mind that a lot of them will converge on this process of helping the phagocytes sense, engulf, and digest microbes faster, better, and more potently. And so many microbes, especially those that cause disease effectively, specifically evade this process. And in doing so, actually can be quite successful. That's how important this singular process is in host surveillance. And so how does this work? There are so many examples of successful microbes that cause active infections that evade phagocytosis. So for example, Streptococcus pneumoniae, which is an important cause of pneumonia, has figured out this way where it will coat its entire surface with this glycopolysaccharide coat, basically a big sugar coat that it puts on the outside of its surface. And by putting this sugar coat, so here it is in green, this is your S. pneumoniae without the capsule, and then this green now is what it looks like with the capsule. Even though underneath the capsule on its membrane, it still has sites that will be readily bound by either antibodies in the fluid or other host factors that will then coat this bacterium. By virtue of having this thick sugar coating, those molecules can't gain access to those targets. And so it's basically almost like a cloaking device. It's occluded those molecules that are targeted by those hosts, those secreted host proteins. It's occluded them with this thick sugar coat so they can't gain access. And so whereas S. pneumoniae that doesn't have that capsule coat on its outside is readily coated with antibodies or, for example, complement or other factors made by host cells, which are then in turn recognized by the phagocyte, allowing the phagocyte to engulf the microbe, eat it, digest it, kill it. None of that process happens in S. pneumoniae that is encapsulated with its polysaccharide coat. And so it becomes effectively invisible to, well, not completely invisible, but largely invisible to the immune system by virtue of this thick outer polysaccharide coating. So this is a pretty effective strategy to evade engulfment. So Staph aureus, I mentioned Staph aureus is a microbe that is pretty ubiquitous on the skin of most people, and it causes some pretty serious infection, it's skin infections in particular. Staph aureus makes a virulence factor known as protein A. And so protein A is pretty clever. What protein A does is it doesn't keep antibodies or other things from coating the surface of the bacterium. So remember, coating with the antibodies will trigger phagocytosis by phagocytes. So it doesn't keep them away, but instead it turns around their orientation. And so it binds to antibodies on the stalk side. So antibodies are these Y-shaped molecules. And it's important to know that the way the antibodies are supposed to work is that at the tips of the Y, so here and here, those are the ends of the antibody molecule that bind its target. So the microbe is its target. And then the long stick-shaped part of the molecule, the stem of the Y, is the part that will actually recruit and engage and activate the phagocytes. So this is the proper orientation of an antibody, and this allows it to do its job of coating the microbe and triggering phagocytosis. But Staph aureus is super clever. So it doesn't keep the antibodies away. What it does, it turns them around. And so it binds to the Y end, the stem end of the antibody. So this is the part that was supposed to engage the phagocyte. And it binds it in that direction. And then it turns the antigen binding end of the antibody, the part that was supposed to bind Staph aureus, outwards. So now it inverts the orientation. And so effectively, it's coated itself with antibodies that are facing outward, and they can't at all gain access to the receptors that are on the phagocyte that would trigger phagocytosis. So it's almost using the host antibodies as a cloak, but it's turned them around in an orientation where it can't engage the phagocyte to do its job. So also quite clever, and this will help it pretty effectively evade phagocytosis as well. So other organisms, and Mycobacterium tuberculosis is one of the best at this, and this is an example of why it is so successful as a pathogen. So Mycobacterium tuberculosis does not evade the phagocytosis part. In fact, it likes to be in the phagosome. So Mycobacterium tuberculosis will be engulfed by a phagocyte. It's its favorite host cell. But what it does instead is it inhibits the phagolysosomal fusion process that would otherwise create a deadly environment for the bacterium inside that phagosome. And so by doing that, by inhibiting the fusion process, it can continue to grow quite happily inside the phagosome, but it isn't killed because that environment never turns acidic. It doesn't acquire reactive oxygen or reactive nitrogen species. It doesn't acquire antibacterial proteins. And so in this way, Mycobacterium tuberculosis is quite happy to have these immune cells these phagocytes that are recruited to the lung that are supposed to be protective, but instead it sets up shop and lives inside of them. And so this is how it does it. And again, this strategy is so effective that this is really one of the world's deadliest pathogens for all of humanity. Humans are its primary host. So this is a tremendously effective strategy. And then the last, strategy we'll talk about today is one of classes of organisms that also do not mind being taken up by phagocytes. But again, they are able to evade the phagosome prior to it becoming a phagolysosome compartment. And they do this by actually, some of them will puncture the membrane of the phagosome and escape into the they'll translocate out of the phagosome and enter the cytoplasm, and then they actually live in the cytoplasm of the host cell of the phagocyte. So species that do this really well include many Listeria species. So Listeria is a foodborne pathogen. Typically, it does not infect many immunocompetent hosts, but is a substantial concern for immunocompromised individuals that have a weakened immune system. And it's one of the most successful things that it does is it will be happily engulfed by the phagocyte, but before the lysosome fuses with the phagosome, it breaks out into the cytosolic region. And now it actually has access to a very nutrient-enriched environment. I mean, all of the host cell's nutrients are in the cytoplasm. So all the vitamins, cofactors, energy sources are immediately available to these organisms, and so they can grow quite readily in these compartments. What are supposed to be deadly immune cells become the host for these different pathogens. Okay, so the last two classes of host defense evasion I'll talk about today include secretion of proteases that digest the IgA antibodies that are present in mucosal secretions. So as I mentioned, many of the mucosal organs are coated in very thick layers of mucus, which is really comprised of proteoglycans and mucin and all kinds of other sticky reagents that exist in this environment. And that mucus layer, or the mucin, really helps protect the underlying epithelial barrier from pathogen adherence and entry. It traps the microbes, and then they can be swept away by different physical mechanical processes like cilia or peristalsis. In addition, there are many different types of B cells that exist in these mucosal organs right underneath the epithelial border. So they live kind of right in that subepithelial tissue space. And they secrete massive amounts of an antibody known as IgA. And it's a very special type of antibody that is almost always located in mucosal secretions. It is secreted across, so once the B cell secretes IgA, it actually binds to the epithelial cell, but from the so you can think of, if this is an epithelial border, the IgA is secreted by B cells over here. It actually is taken up by the epithelial cells that form the border and extruded onto the other side, the luminal side where all the mucus is. So the B cells aren't on that side of the border, but the IgA does get there. And so the IgA now becomes trapped, or not really trapped, it binds within that mucus layer. And it is actually very effective at capturing and coating microbes, which can neutralize them. It can help them be taken up by phagocytes that are surveilling, or importantly, can just help them be swept out in the mucus as well and inactivate them. So not surprisingly, a number of microbes that like to gain entry and live in mucosal spaces produce classes of proteins known as secretory IgA proteases. And effectively, what these proteases do is they chop the IgA up so that it can't do its job anymore. And so this is an important class of proteases that help evade IgA coding and detection and a lot of the other processes that would normally keep these microbes inactivated in the mucus layer. So examples include Neisseria gonorrhoeae, which is the organism that causes gonorrhea in the urogenital tract. Streptococcus pneumoniae, which causes pneumonia in the lungs, are two important pathogenic organisms that make pretty large amounts of secretory IgA proteases, and these are virulence factors. And then the last thing we'll talk about today is this concept of antigenic variation. So antigenic variation is particularly important for organisms to evade the adaptive immune system. The reason that cells of the adaptive immune system take so long to be activated is partly because they need to be receiving the correct and prolonged inflammatory signals to let them know that there's an infection there. But the other reason that they take so long to become activated is that both B cells and T cells, those are the important immune cells of the adaptive immune system, recognize very specific molecules on the microbe, as in a very particular part of a very particular molecule. And they do this by recognizing either T cells recognize little peptide pieces, a very specific peptide sequence in a very specific protein in the microbe, or B cells recognize three-dimensional surfaces, and they're so sensitive that it has to be the exact match, which is usually a very specific part of a very specific molecule. So all of this specificity is great because B and T cells are important for very targeted specific attacks and are important. That's why when you're vaccinated against a very specific organism. Not all organisms, a very specific organism, and you're engaging B and T cells in this process. But of course, the flip side of that is all the organism needs to do to evade detection is change that very specific part of that very specific molecule, and it no longer can be recognized by the B and the T cells. And changing that very specific part of that very specific molecule can literally be accomplished by a point mutation. That's how specific it is. And lots of organisms in the course of replication undergo mutagenesis. Bacteria are great at this, viruses are great at this. So one point mutation can be sufficient to evade detection by B and T cells. Yes?
So it's happening by chance, right? It's not that the bacteria can purposely go through mutagenesis.
Awesome question. So both happen. So some organisms, it's just a random mutagenesis-driven process. Other organisms have devised these super clever ways to, there are lots of ways to do this, but a common theme is they'll have a class of proteins expressed in, for example, let's say one big locus, where you have protein A, protein B, protein C, protein D, and all of those proteins are, for example, a pilus on the surface, a pilus that is able to help the bacterium attach to the surface, but maybe has slightly different structural components to it. Tiny differences, so it works the same way, but just a little bit different. And so it will, sometimes randomly, but sometimes at very specific orchestrated steps, will switch from using pilus A to using pilus B. And then at some other point, it will switch from using pilus B to using pilus C. And so you can imagine when it's using pilus A, there's this whole immune response against pilus A that involves pilus A specific T cells and pilus A specific B cells. But then by virtue, so you have this fantastic immune response, you clear most of the organism, everything's going great for the host, and then boom, it switches. And there's lots of ways to control this, and lots of organisms have very clever systems, but now it will turn off A. It's not gone from the genome, it's still there, but it's not using it, and now it's going to use B. And so B is on the surface. And so now there's this 2, 3, 4 week delay where the adaptive immune system needs to become activated and produce enough cells. And that whole delay occurs again. And in the meantime, the organism is growing, replicating, disseminating. So lots of successful pathogens have devised mechanisms like this. So you can have random mutation. Viruses are great at random mutation. And then there are lots of other organisms. Gonorrhea is another example that uses this pillin strategy. Trypanosomes have this really clever way of switching out cell surface molecules that are just slightly different in architecture also on their surface, and boom, you can't see them. And you have to wait 2 or 3 weeks for the B and T cells that are specific to those proteins to replicate and gain enough activation in the periphery. So many organisms that partake in antigenic variation, what you see over time in the host is waves of an immune response, where you have an immune response amplifying, reaching this peak. Often at the peak, the pathogen numbers are very low because the host immune response is aggressive and doing a great job. And then boom, the pathogen switches to a different surface protein. Usually it doesn't alter its function either. And the immune system becomes useless. It goes away, it dies off, and now you have to wait for a second class of T and B cells specific to that other protein to really ramp up. Pathogen is slowly eradicated again or cut down, and you just do this wave after wave after wave. So there are a number of really successful pathogens that cause notable diseases that use this strategy. Yes?
So when we talk about the first method of creating phagocytosis, we talk about using that outside capsule.
The outside capsule. This one, yes. It's a little different because phagocytes they don't recognize very specific things. Phagocytes, the receptors on phagocytes recognize super broad classes of molecules that are associated with pathogens. So for example, there are classes of receptors that will recognize lipopolysaccharide, right, which is part of the outside of the bacterial coat. It's not really found on host cells. So it's a bacterial component associated with bacteria and certain classes and types of bacteria in particular. So the specificity is a lot lower, and those receptors don't stop recognizing it, nor does the bacterium stop producing that target, but instead it just basically covers it up with a thick layer of polysaccharides. So both the target and the receptor are the same. There's no difference there's no variation. They can't get access to each other because you have this thick polysaccharide coat that's in between them, if that makes sense. Yeah. So it's a little bit of a different strategy. All right, so with that in mind, let's take a break. And I think we're doing okay on time. 4:30. Let's do like a 15-minute break, 10, 15-minute break, and we can start with the last half. Do you want me to do pause? This guy hit it? Great. So for the second half of the lecture, we're going to talk a little bit more specifically about the different types of toxins that are produced by bacteria and how those toxins can interface with host cells and cause a lot of the disease states or disease manifestations you're used to seeing from a clinical perspective. And then we'll talk about a few different pathogens that are highly clinically relevant and ubiquitous. These are pathogens that you either do or should know about, and many of them are targeted by very successful vaccines that are required during childhood in this country. So first, let's talk a little bit about bacterial toxins. So what are bacterial toxins? These are typically proteins that are specifically produced by the bacteria to harm host tissue or trigger destructive biological activities in the host. Some toxins and other bacterial virulence factors have other roles for the bacteria in supporting their life cycle or things like that, but by and large, a toxin, the entire goal is to target a host cell and harm it, usually kill it. So there are two broad classes of bacterial toxins you'll hear a whole lot about if you stay in the microbial pathogenesis field or in the immunology field. Those include the endotoxin classes of toxins and the exotoxins. So for today, I'm only going to briefly mention what endotoxins are, though if you ever take an immune class, you'll hear a ton more about these. And then for the next few slides, go more deeply into exotoxins. So the big difference is endotoxins stay associated with the bacterium. Usually they're membrane or envelope components or proteins that actually stay embedded in the cell wall or the cell membrane of the bacterium. And exotoxins are secreted. They can be directly injected into target cells, although a lot are simply secreted into the extracellular fluid, like the blood or the other fluid drainings, and by virtue of diffusion find their targets on host cells. So endotoxins, as I mentioned, largely stay associated with the bacterium. They are almost all intrinsic cell envelope or some cell membrane components. They serve their really big role from the host standpoint is to serve as alarm triggers to the immune system. So immune cells express a huge diversity of different receptors that recognize many of these different bacteria-associated molecules that are part of the cell membrane. And not surprisingly, that's because the host immune system has evolved to detect microbes. That's kind of one of the most fundamental functions, so it detects them so it can kill them. And so often what it's detecting are these endotoxins. And they're called toxins not necessarily because they're intrinsically toxic to the host, but because what they do really, really well is activate host immune cells. And what many host immune cells do once they're activated, so one thing is if it's a phagocyte, it'll eat the microbe. So that's a great thing for the host, bad for the microbe. But the other thing that host immune cells do when they've become activated, particularly by these endotoxins, is they secrete copious amounts of inflammatory mediators, especially inflammatory cytokines. And those cytokines are really important for the inflammatory response that you experience as pain, swelling, redness, and heat, all of which are designed by the host to hinder bacterial growth or microbe growth and help cells of the immune system. So fever, for example, or hot tissue immune cells actually function slightly better at higher temperatures, whereas most bacteria fail to replicate well at slightly high temperatures. So all of those responses is caused by the secretion of these inflammatory factors, mostly known as cytokines. These are the protein ones that are secreted. But some of them, for some of these cytokines, the production is so over the top, and particularly if it's occurring systemically, not just in the tissue, but all over the body. This happens a lot when bacteria have entered the bloodstream. It actually puts the body in a state of shock. So for example, septic shock is typically caused by a bacterial cell wall envelope protein or molecule known as LPS that's being carried on bacteria that are bloodborne or actually being shed just because bacteria are naturally dying and kind of their cell wall components are being released, sloughed off is the right way to think of it. But it's happening systemically. It's activating so many immune cells so highly, they're producing just this swamp of cytokines, and it basically shuts the body systems down in a state of shock. So septic shock is really caused by your immune system that's gone haywire. It's too activated, it's producing too many cytokines, and they're not being held locally, but rather hitting all the organs. And so some of the outcomes of the immune system that are usually good for the host, like high fever. Another thing that inflammatory cytokines do is they'll actually cause microclotting in an infected tissue, and this kind of helps wall off the infected site. Those things now actually are happening in organs that are our vital organs supporting the body system, like your heart, like your lungs, like your kidneys, like your liver. And so it gets too hot, you have microclotting in all of your vital organisms, and eventually you have organ failure. Your body can be so hot you have disruption of brain function, and all of these things ultimately can lead to death. And they're all caused by the fact that your immune cells are just simply too activated by these toxins being produced on such large scales and all over the body. And so that's an example of how endotoxins harm the host. They're basically hijacking a normal host immune system, but cranking it up so high that it kills the host. There are lots of examples of endotoxins, and different classes of bacteria tend to utilize different classes of endotoxins. So gram-negative bacteria are known for LPS in their cell wall, and LPS is a fabulous endotoxin. In fact, for many years, the field has used the LPS, which comes from gram-negative bacteria, interchangeably with the word endotoxin. So if you read old papers and they're talking about endotoxin, they're actually talking about LPS, which is a specific molecule that's recognized by a specific receptor. But gram-negative bacteria make other endotoxins in addition to LPS, and gram-positive bacteria have their own classes of endotoxins. These include peptidoglycans, teichoic acids, things like that, which again are normally very good at triggering immune cell activation locally and for the host advantage, but when they become systemic or just too much, when bacterial outgrowth has just become overwhelming, that response gets out of control and it will itself kill the host. Okay, so that's all I'm going to say today about endotoxins, though if you do immunology, you'll hear a lot more about them. So for the rest of the lecture, we'll talk about exotoxins. And so exotoxins have key roles in producing a lot of the disease manifestations that you would associate with typical pathogens. These are proteins that are made by all kinds of different bacteria, gram-positive, gram-negative, acid-fast, you name it. There are really three major classes of exotoxins. These include the AB type toxins, which I'll talk a lot more about, and these were the type of toxins, that were well understood the earliest. So a lot of literature and research has been focused on AB type toxins. Although we now know that bacteria produce other classes of toxins, exotoxins, that include membrane disrupting toxins and superantigen toxins. And by virtue of the fact these were sort of studied later and less in depth, we're still getting a clear understanding of all the different types of bacteria that make these and how they act. So we know a lot about AB type toxins and a little bit less about the other two, but some. So as I mentioned, many of the clinical manifestations of diseases you'll hear about, especially the more horrific ones, are being caused by toxins that are produced by the pathogenic bacteria. These include a lot of the diarrheal diseases that are associated with poor water hygiene or poor food hygiene or food contamination. Though some of the toxins that we have now sort of studied well enough, we've figured out ways to harness them for clinical and medical uses. And this includes and I'm blanking on the name, the toxin that is Botox, the botulinum toxin that is injected into skin to help dissipate wrinkles, but basically all it's doing is paralyzing the facial musculature underneath so that you're not squinting up your face and causing wrinkles anymore. So that's a cosmetic use of it, but Botox actually has other non-cosmetic uses that are really important, including there are a number of disorders that are the underlying cause is spastic muscles that are overly tensed and fail to release, including cross eyes, strabismus is a muscle connected to the ocular system that if you inject just the right way with Botox, you can relax it, and it actually helps treat the disease. So Botox is a great example of a toxin that actually is tragic when it's being produced by the bacterium in a pathogenic state, but if we can harness it in just the right way, we can use it. Okay, so the types of toxins I mentioned, there's the AB type. This is the type we'll talk the most about today. And the AB type is comprised in general of two components, the A component and the B component. And in most cases, the A and B component are actually produced as one big long polypeptide, and then they're processed to become two separate pieces, two separate, if you will, protein components, but they act together as a pair. And the A component is the part of the protein that is enzymatic, so it's the functional end of the toxin. The B component is the part of the molecule that is important for binding the target host cell. So the B component often confers specificity. Some toxins will actually enter almost all host cells. Other toxins are very, very particular about the types of cells that they're targeting, and that's by virtue of specificity in the B component of the AB toxin. So an easy way to remember that is A is for activity, B is for binding. And there are kind of two versions of these AB type toxins. There are the simple types, and those are comprised of one A subunit linked to one B subunit, so it's a 1-to-1 ratio. There are also complex AB type toxins in which you have one active A subunit attached to many different, or many either multiple copies of one B unit or actually multiple copies of different B units. So the B unit can be the same or different, but it'll be attached to multiple copies of the B. And we'll talk about two examples of those as we go through today. So those are AB type toxins. There are also the membrane disrupting toxins, and these typically fall into two categories. The passive channel complex, and literally what it will do is form a pore or a channel in the host cell membrane. And often in doing so, what it's doing is disrupting the ion gradient of the extracellular space to the intracellular space. We'll talk about why that leads to death of the cell in a minute. And then there are the phospholipase genre of membrane disrupting exotoxins. And these also destabilize the membrane, but they do that by actually chopping off the phosphohead of the phospholipids in the cell membrane. It destabilizes the membrane integrity and just kind of makes the membrane overall weak and leaky. And then that also will lead to lysis of the host cell. And then the last class is a super specialized class of toxins. known as super antigens, and these act by targeting cells of very specific types of cells of the immune system, and again, just overactivating to the point where they go haywire, and you have this systemic flood of cytokines and inflammatory signals that kill the host, much like an endotoxin, but the mechanism is much more specific. Yes?
Is the point of these toxins to like actually kill off the host cell or just the don't they eat the host cell?
Depends on, so you hit on a really interesting point of a successful pathogen will kill just the right amount of hosts to get its needs fulfilled. So it doesn't really care if the host lives or die. A lot of pathogens don't just live in one host. They rely on serial transmission for that. So it can kill hosts, but it can't kill them so quickly they don't have a chance to transmit. That's the key. So it's the more successful ones are only killing at the right rate, so that there's a constant reservoir and there's enough cross-person transmission to keep the organism alive. Also keep in mind a lot of pathogens they're not obligate to the host environment. So a lot of pathogenic organs are naturally soil-dwelling organisms, and they don't need a human host to live and carry out their life cycle. They live in a lot of different environments. We'll talk about Pseudomonas aeruginosa, for example, which can basically live everywhere: water, soil, humans, animals, you name it. It's incredibly versatile in its metabolic requirements. So it really depends on the kind of pathogen. Clostridium botulinum, the one that causes botulism, is also mostly found in the environment. So bees carry it, it gets attached to pollen, and then bees carry it on the pollen, so you often get the spores and honey, but it doesn't need a human host at all. We're irrelevant. So again, it just kind of depends on what its host requirements, its metabolic requirements are. But if it does require a host, and a human host, it will only be successful if it's killing slowly enough that it can be transmitted. So I guess you have to kind of keep that in mind. It doesn't always need us. It doesn't always care. I mean, not in a anthropomorphizing sense, but just really in an overall survival, we could be possibly irrelevant. We could be a source of nutrients when it's convenient or not. Okay, and so super antigens are acting by overactivating the host immune response. So here's an example of really an oversimplified cartoon of what simple and complex AB toxins look like. So you can imagine the binding of a simple AB toxin, the binding subunit is here in B, and it is especially prior to activity directly linked, usually in a covalent bond to the A subunit. Often once the AB toxin gets to its host or even gains entry to its host, these two will be released from each other, but not always. They can actually gain entry and stay together. And a complex AB toxin looks, for example, something like this, where you have the active subunit bound to multiple B subunits that could be the same or different from one another. So how do A subunits gain access to the cell? So by and large, a toxin, in order to be effective, needs to get inside the cell. And the reason it does that is almost all toxins are going to target enzymes that are very important for the cell on the inside of the cell. These might be metabolic enzymes, these might be enzymes or proteins that are important for function, but things that are critical to that cell's activity, life cycle, ability to exist. So it needs to get inside. And it usually does this in one of two ways. It can be endocytosed, which is a process quite similar to phagocytosis, though endocytosis can be mediated by lots of different immune cells, and it does not result in a lysosomal compartment. But endocytosis as a process is much like the early phagosomal process, where the membrane of the host cell basically engulfs the entire complex, the entire protein complex, and takes it interior to the cell. And early on, that compartment will be comprised of what is effectively extracellular fluid, and then over time it can shift to be more like the cytosolic fluid in similarity. And usually that toxin, once it's inside the cell, still has to get out of that vacuole and into the cytosol, but it's taken up into the cell through endocytosis. And then the other mechanism is a process known as translocation. And this is typically a straight injection system. So some pathogens, bacteria, encode these secretion systems that effectively they're on the outer surface of the cell wall or some membrane. They'll dock on the host cell and they create a very tight channel that links the bacterium to the host cell. So almost a pore that directly links the cytoplasm of the two organisms, and then it'll inject proteins into the target cell. So it's like a syringe, if you really want. That's what it looks like too, microscopically. So you can directly inject the A and B components into the cell where it gains access to the cytosol that way. So the typical steps of how a cell becomes intoxicated with a toxin, the bacterium will adhere to the cell surface. Sometimes toxins are secreted at one site and act distally, and I'll give you a really cool example of one that does that, but often they're happening locally. The toxin is produced and often secreted either into the extracellular space, if it's going to be endocytosed, or secreted in a way it's going to be injected directly into the cell. It is often synthesized in a proform where it's not active at all. It can't do much of anything until it is processed in a way that releases an active form. So you could think of something like an ABC subunit, and the C needs to be cleaved off before you get the proper AB toxin existing. Although some toxins are secreted directly active. They don't need any processing, so this depends. You now have your active toxin, which needs to gain entry to the cell. A lot of toxins will bind to receptors that are actually on the target cell membrane. This can be a ubiquitous receptor or a cell-specific receptor. By virtue of the binding event, it could be this complex can be taken up into the cell by endocytosis, as I mentioned, or it can gain entry. This can be an injection entry. This can be via formation of a pore in the membrane. But somehow, depending on the toxin, the A component needs to get into the cytosol of the cell. And then once it's in there, it will catalyze whatever enzymatic reaction it's made to catalyze, and this almost always leads to some sort of inactivation of a critical host machinery or protein where the cell will then die or do all sorts of other activities it's not supposed to do. So this is the basic schema of how the AB toxins work. Not surprisingly, a vast array of pathogens you're used to hearing about as disease-causing agents make different toxins. So these include organisms like Bacillus anthracus, which makes the anthrax toxin. You guys have probably all heard about that. Bordetella pertussis, which is a causative agent of whooping cough. Few of you probably will have experienced because there's a really great vaccine available, but prior to the vaccine or anyone unlucky enough to catch it, it's a terrible, terrible lung disease that's just horrifying to experience. There are the Clostridium species, as mentioned. Clostridium botulinum makes the botulism toxin, the Botox toxin that we use cosmetically and medically as well. Clostridium tetani, which causes tetanus. So most of you will probably be vaccinated against tetanus as well. But tetanus is the disease that's associated with that phenomenon of lockjaw I mentioned, where your muscles become so spastic that your jaw locks down and you can't eat or drink. And in some people, the spine, it will be so spastic that the spine will curve around in this very deformed, painful way. And it's nearly always deadly. It also leads to respiratory arrest because of spasming of different muscles as well. So super deadly disease, but fortunately we have a good vaccine. There's the diphtheria toxin, we'll talk about more later. Many different strains of E. coli are the reason that they cause diarrheal diseases when they do. So you know there are E. coli everywhere in the environment, but some of them are particularly strains that cause disease symptoms, especially diarrhea. That's because they're often making these toxins. So the strains that make the toxins are the ones that are disease-causing, and the ones that don't are fairly benign and have commensal functions in the organism. Pseudomonas aeruginosa, we'll talk a little bit about this organism. It is particularly prevalent in cystic fibrosis patients. It invades the lung. It actually can live in all kinds of tissues, but it loves the lungs of cystic fibrosis patients and causes pretty nasty lung disease and infection in that setting. Shigella, cholera it's the cholera toxin that causes cholera disease. So these are all organisms you've heard of, and most of their disease manifesting elements are about their toxin production. So also in that vein, it's not surprising that a lot of the vaccines that we develop against these organisms, many of which are actually pretty effective now in recent decades, are actually targeting the toxin. Not so much the organism, but if you can neutralize the toxin, then the organism becomes non-disease causing. And so there are some great vaccines that specifically act by neutralizing the toxin itself. Okay, so what are these, the activity or the A component of these toxins doing inside the cell? As I mentioned, they have a lot of different functions. So it really depends on what particular class of enzyme they are. But a very ubiquitous class of enzymatic function of toxins is to act as NADases. So NAD is the nicotinamide adenine dinucleotide factor. NAD is what it's always called. Is a particularly crucial metabolic cofactor in a whole bunch of different metabolic pathways. All kinds of energy-producing pathways in your host cell need this cofactor in order to operate. And so NADases use the NAD molecule as a source of a substrate. And so what they do specifically is they tear off this ADP ribosyl piece off of NAD, which is everywhere in your cell, a great source. And they'll transfer that tag to another target protein. And by virtue of transferring that, tagging another protein with this ADP ribosyl protein, they inactivate it. It's a big, huge chemical tag they put on a protein and basically will shut down, a lot of times it just shuts down the action. And so by doing this, depending on what that specific target protein is in the host, you can shut down all kinds of important host functions if you choose the right target. So we'll talk about an organism that makes an NADase toxin that stimulates cyclic AMP stimulation or production, overproduction in cells. We'll talk about another one that actually uses that ADP ribosyl protein tag to inactivate elongation factor 2. And elongation factor 2 is one of the most crucial, critical, central players in protein synthesis. And so if you as a cell don't have functional EF2, you don't make proteins and you die. And so it's a pretty elegant way to shut down a cell. You just have to choose the right target. So we'll come back to specific examples of specific toxins when I talk about the organisms that I want to go through with you. But suffice it to say, those are generic examples of how AB toxins work. So there are also, as I mentioned, the membrane disruption version of toxins. And so those typically act in one of two ways. If they are pore-forming toxins, they will actually usually insert right into the phospholipid bilayer of the cell membrane. Yes?
I was just wondering, for this one, does it have any relation to how cancer cells can like form flux proteins to pump out drugs?
Actually, yes. So a lot of them are pore-forming proteins. They don't necessarily have to be as complex as an efflux pump. An efflux pump is usually a little bit more sophisticated machinery, and it's sensing ion concentrations on one side of the membrane versus those on the other, and usually changing conformations. I would say this is more of a rugged version of a pore-forming complex, in that it acts a little bit more just ubiquitously. It inserts, and in a sort of unregulated way, by virtue of inserting and basically creating a channel in the membrane, it's disturbing the fact that the outside of the cell is very low osmolarity and the inside of the cell is very high osmolarity. And as soon as you create an open channel between systems that have differences in osmolarity, they immediately go toward equilibrium. And so what does that mean for the cell? It means a whole bunch of water in the extracellular fluid rushes into the inside of the cell. It swells, and it pops. And so they're working in a very crude way. They're basically punching a hole in it and letting osmolarity, the natural effects of osmolarity, take over and pop open the cell. So that's the crude way. So they're not sensing as well as a good efflux pump is. But the idea is similar. So that's one generic mechanism of a lot of these membrane disrupting toxins. The other flavor are these phospholipases, and a whole bunch of different pathogenic bacteria make phospholipases. I would say the majority. And they actually act by just clipping off the phospho head of the phospholipids that form the cell membrane, the bilayer of the cell membrane. And by clipping off those phospho heads, you're destabilizing the entire membrane structure, and over time the membrane of the cell becomes weak and leaky and loses integrity, and again you'll get holes and the cell will swell and pop. So they're both acting to destroy the membrane integrity and its key function in keeping a very tight and distinct ion balance on one side versus the other. Okay, so you have the AB toxins, you have the membrane disrupting toxins, and then the last class of toxins we'll talk about today are those that are known as superantigen toxins. And so in order to understand how superantigen toxins work, you need to understand the normal process that they're hijacking. So the normal process that they're hijacking is a process known as antigen presentation in the immune system. And if any of you will take immunology or are, this is a pretty cool pathway. But what happens in the antigen presentation pathway is you have an antigen-presenting immune cell, and a lot of different immune cells can do this, but dendritic cells are the best. You don't need to know what that is if you're not studying immunology. And what they're doing is they're taking peptides, so peptide antigens, they're loading them onto this surface protein known as MHC class II. And all MHC class II is basically a great big holder that they'll use to present, almost like a velvet pillow, to the T cell. And I mentioned to you T cells are really important parts of the adaptive immune system. And this is part of the reason it takes so long to get a T cell activated. So a T cell will only bind this antigen peptide presented in this MHC2 if it specifically recognizes it. And so this gets down to the specificity. About 1 in 10,000, or even 1 in 100,000, or 1 in a million for some antigens, you actually get specificity where that T cell exactly recognizes that tiny piece of peptide in that exact MHC molecule. So very, very few T cells will actively engage in this recognition, but if it does, it becomes activated. And then, as I mentioned, an activated T cell ultimately goes on to do all sorts of really important things. But they're few and far between where this is a good match. Most of them, it's not the correct match and nothing happens. So under normal circumstances, if it's the right match, the T cell will become activated. It will make a cytokine known as L02, which acts on itself and other immune cells to activate them, cause proliferation. An activated T cell will now engage B cells. By virtue of engaging those B cells, it will activate the B cell. That B cell will now proliferate and start to make antibodies. And so this is the key interaction that drives an adaptive immune response. And it is slow, and everything has to be perfect. And if it's not, it doesn't happen. So this happens infrequently, but when it does happen, it's incredibly powerful. So superantigens do something, they sort of cheat. And what they do is they've developed a way to bind the MHC and the T cell receptor in a way that is completely antigen independent. So it doesn't even matter what that peptide is in there, and it doesn't matter if that T cell receptor is specific for it. And so it almost links them together into a false signaling complex. The same sort of orientation you would get if this were a good match, but it basically locks it in place. And this happens to most T cells. Well, most might not be, but incredibly, incredibly large numbers of T cells. So about 1 in every 5 will be actively connected like this. And the T cell doesn't know that this is not a proper recognition. It just realizes it's engaged in binding MHC, and it becomes super activated for super long periods of time. And you get all of these T cells in your body all over the place, making huge amounts of IL-2, doing all the things that T cells do, but on overdrive, on hyperdrive. And this leads to a cytokine storm in your body and septic shock and usually death. Because again, it's this notion of the immune system's very powerful, but you want it localized and specific. When it's everywhere and out of control, it's deadly. So this is what super antigens do, and they're toxic because they kill the host with its own immune system. So that's it. That's all I will talk about now for toxins, and then for the last, I guess, maybe 15, 20 minutes of the lecture, we'll go into some examples of some specific bacterial pathogens that are responsible for notable diseases. So the first one I want to talk about is Corneobacterium diphtheriae, and this is the causative agent of diphtheria, which for many decades, for eons, and then up until only recent decades, we did not have a great vaccine for it, and it is a pretty deadly pathogen. So the Corneobacterium diphtheriae is a gram-positive bacillus. It is non-motile, and this is what it would look like if you put it in a dish and looked at it under a microscope. Not particularly exciting looking, and it doesn't move or do anything cool. Nevertheless, it's really great at colonizing your throat and nasopharynx, and it's transmitted from host to host, not surprisingly, by inhalation. Either sneezing, coughing, anything that would allow droplets of fluids containing diphtheria bacteria to be transmitted from one person to another. It is non-invasive. It stays in the throat or your nasopharynx, and all of the damage it does, and it does some pretty serious damage, it does from that location. So it's not moving deeper into the body. It's not moving into the bloodstream. It's not a disseminating bacterium. It stays localized where it is. And it forms this super gross, thick gray mucoid coat over the interior of your tonsils and your upper respiratory airway tract. So you can kind of see it in this person. The tonsils are right here. You have the uvula, and you see this gray, gross, slimy looking coat. And that is pretty indicative of diphtheria. And even better, that coat is not just sitting on the surface of the epithelium of your nasopharynx. It's actually directly bound. And so one of the really great ways to know you have it is if you try to pull the membrane up, like a doctor tries to, it bleeds because that's how tightly adherent it is to that layer. So it's pretty gross. And in this mucoid coat, you have fibrin from the host. You have a whole bunch of bacteria. You have all kinds of inflammatory cells that are trapped in there trying to get rid of it, but not actually having any success. And then from this location, it secretes toxins that become bloodborne. So it stays there, but the toxins go everywhere. And it is the toxins that are secreted by diphtheria that do all of the damage in the disease. And many individuals, if they're not treated really quickly, or even if they are, the result is coma or death. So one of the important organs that is targeted by diphtheria toxin is the heart. And so early on in the disease, you get an irregular heartbeat. Over time, this worsens and can lead to things like coma and death. It also affects the peripheral nerves, and one of the manifestations of that is difficulty swallowing, which has all kinds of implications for the host as well. And it's not just because it hurts to swallow, it's because the muscles that you need to swallow stop acting properly. And all of that's mediated by these toxins that are being secreted at within this localized mucoid coat of bacteria and gunk. So what is this toxin? Diphtheria actually secretes a few toxins, but the one you will know the most about, and that is responsible for a lot of the clinical damage, is the one that is known as the diphtheria toxin, or simply tox. It was well known early on and got a pretty straightforward name. Tox is iron regulated, so it is produced, it is upregulated under iron limiting conditions. And it has been proposed that one of the things it might be doing is actually, by killing target host cells, it's releasing internal iron stores. It probably has that role and all kinds of other roles in it. That might not be its essential function, but it's a helpful function. It acts by binding a host receptor known as the heparin binding EGF precursor receptor. So this receptor is actually expressed on most human cells. But it is particularly highly expressed on the heart cells. So that explains some of the reason why a lot of the clinical damage is is occurring because of destruction on heart tissue. Engaging, so the AB toxin is made as a linked polypeptide of where the A and the B are connected to each other. It's a simple, so there's one A, one B. The B binding to the heparin binding EGF precursor receptor triggers endocytosis. At some point in the endosome, the A component is released from the B component. So this is an example where A actually gets released and is separate. It ends up in the cytoplasm by a process that is not entirely perfectly well understood. And then in the cytoplasm, it is one of those toxins I mentioned that acts as an ADP ribosylation enzyme. So it takes the ADP ribosyl group off of NAD, which is everywhere, and it transfers it to the host proteins. And its best and most well understood target is elongation factor 2, which, as I mentioned, is critical for protein synthesis in a host cell. By doing so, it is so effective, it shuts down host synthesis of proteins and the cell dies. And so just to give you an idea how effective this toxin is, a single A chain, one molecule, can kill a host cell. So that's pretty impressive, especially when it's targeting the heart. And so you can, I think, infer why this is such a deadly pathogen. It's very good at what it does. So that said, we've done a decent job at using intellect and hard work to develop a good vaccine against diphtheria. And most individuals will have been vaccinated very young. So diphtheria is actually a component of the triple DTP or DTaP vaccine that you'll get a number of shots and booster shots throughout childhood. And it is the part of the trivalent vaccine, the D component is for diphtheria. So that's what this is. And so as a result, actually, very few deaths worldwide. This is how good this vaccine is, about 5,000 of those. And so most of that is about treatment with the vaccine and preventing it. We have developed antitoxins and antibiotics as well that are somewhat effective. But by and large, the way that we've controlled this pathogen is just simple, straightforward vaccination, and it's great at doing what it does. So that's diphtheria. Another one that you will be most likely vaccinated against as a child or will have been is Bordetella pertussis. This is a small gram-negative coccibacillus, and so here it is in a dish, just to give you a sense of what it looks like. It is a bacterium that is known as nutritionally fastidious, and so what does that mean? It means it has very, very specific nutritional requirements and not a non-versatile metabolic system. So it only grows under very optimal conditions. It manages to find them, but they're not everywhere. And so it's often, if you grow it in a lab, you use blood agar plates, which are very rich in a whole bunch of different nutritional components. So this thing isn't everywhere. It's actually super specific. It is a lung and respiratory, really much more of a respiratory pathogen that targets the respiratory system. It's spread by aerosols and direct contents, and it colonizes the respiratory system from the point of entry, so the nasopharynx region, all the way down to the branches of the bronchioles that feed the lung. So kind of the upper respiratory tract. It rarely goes into the lungs themselves or deep into the lungs. So it's really, really upper airway. And so even though that is a relatively harsh environment for most bacterium, lots of ciliated epithelial cells, lots of mucus, lots of things that are constantly clearing your airway to keep it open. Bordetella pertussis has developed some great adherence mechanisms that allow it to gain adherence, and colonize anyway. And its best mechanism is the fact that it has very high specificity for those ciliated cells that are supposed to be getting it out of there. And so it will bind the surface of the ciliated cells, the ciliated epithelium in your airway. It will start to multiply, colonize right there on the surface of those ciliated cells, and they will die. And so enough of these die, there's nothing that's actually pushing it out anymore. So even though it becomes trapped effectively, and then it can go on to do what it needs to do to survive. So as I mentioned, rarely causes pneumonia or deep in the lungs, and almost never gets into the bloodstream. So it stays where it is. So pertussis, which is the disease caused by Bordetella pertussis, is a nasty one. It is often referred to as whooping cough. So pertussis, although the vaccine is very effective and most people have it, some adults and even some children can still get mild forms of the disease. So it's not that you will never have known someone who had whooping cough, but most likely if they are vaccinating, it'll be a very mild form of it. The non-mild version is quite severe. So it's a pretty long course of disease. It has about a 5 to 10 incubation day where you've been exposed to the bacteria, they have adhered to your ciliated epithelium, and they're starting to colonize and grow, but you are not necessarily experiencing any symptoms whatsoever. Then there's the cataral stage, which is the stage where you start to realize you're sick, you have cold-like symptoms, but again, they're super mild. This is not the kind of disease where early on you're going to the doctor at all. And sadly, this is the phase also where the transmission is beginning. So you're not only are you starting to feel a little ill, you're able to transmit pretty effectively. And even worse, by not going, because the disease in the early stages is so mild, there's a lot of bacterial outgrowth happening before you even know what's going on. It's the late stage of disease where I think most people start to realize something pretty bad. And this is the severe whooping, what's known as the paroxysmal stage or the whooping cough stage. And that's where the host will develop that severe, horrible spastic cough that you get a whooping sound that is caused by the toxins produced by this bacterium. And then people who have whooping cough or whooping cough pertussis, this is a pretty long disease. It takes, if they survive, it's many, many, many weeks or months of being sick. So it's highly contagious. There are still about 39 million cases worldwide and about 300,000 deaths as a result. This is despite the fact that there's a vaccine. In the US, we're very rigorous, especially for enrollment in school, of requiring the DTaP, and it's the AP part of DTaP or the DTP vaccine. So the incidence is low and the disease is typically mild, but it still exists. And so how does Bordetella do its job? And actually, I should mention that, so by the time people realize they have whooping cough, as I mentioned, it's pretty advanced. And at this stage, the antibiotics, though they can be administered clinically and they can they can help limit spread, they actually don't do almost anything to stop the colonization that's already occurred. So at that point, it's so advanced, you're fully colonized and you're going to experience the disease. It takes the edge off and might help limit transmission, but at that point, you're in. You're all the way in, and you're going to be as sick as you're going to be. Okay, so how does Bordetella establish disease so effectively? So first of all, it has a number of different adhesion proteins. Probably the most well-known and well-studied is the FHA protein, or filamentous hemagglutinin. And this is the adhesion molecule that allows it to bind so specifically and so tightly to the ciliated epithelial cells. So this one is really important for letting it establish adherence and then colonize that upper airway tract. Once you get adherence of Bordetella, then it starts producing it actually has a number of different toxins, but two that are well-studied, well-understood, and it produces in pretty high quantities are the adenylate cyclase toxin, which is a simple AB type toxin, and the pertussis toxin, which is the one you've heard probably the most about, which is a complex AB type toxin. And so the adenylate cyclase toxin is an adenylate cyclase. Once it gets into the interior of the cytoplasm of its target cell, it causes that target cell to overproduce cyclic AMP. So usually cyclic AMP is needed in the cell, but once it gets to a certain level in the cell, there's all sorts of negative regulatory pathways that shut down its synthesis. So it's always maintained at exactly the right spot. But this adenylate cyclase toxin gets in there and it actually just creates a cell that can't stop making cyclic AMP. Ultimately, this will kill the cell. And then the other angle that it takes is that pertussis toxin is an NAD ribosylase enzyme, or an NADase, and so it transfers the ADP ribosyl group that we've talked about, this enzymatic function, but it tends to target G proteins in the host, so things like RAS protein. G proteins, and if you've had any cell biology, G proteins do all kinds of things. They do adherence, they do motility, they are involved in all sorts of sort of cell membrane to metabolism regulation functions. So they really have a diverse repertoire of functions in the host cell, and pertussis toxin will shut those down. And so the end result of all of these toxins in particular is that you get death of those ciliated epithelial cells it's bound to. Those die pretty quickly. You get death of a lot of immune cells that get recruited in to try to get rid of it. They die from the toxin, so they're not doing anything. And it's also acting on some of the neuroways in the neurons in the airway system, so that's part of what's driving that really nasty spastic cough that is the underlying feature of the disease. So this is just an example of how the pertussis adenylate cyclase toxin works. So it is this, sorry, we'll just put them all up there. So you have adenylase cyclase, and it's entering the cell. This is the A component of that simple AB toxin that pertussis makes. It itself is able to drive production of cyclic AMP in the cell. In addition, you have the pertussis toxin, the tox part of it, the actual toxin binding G proteins, including a G protein that is important for shutting off the host cell's adenylate cyclase, which drives cyclic AMP production by the host as well. You're also inhibiting a whole bunch of other G proteins, and ultimately you have this massive increase of cyclic AMP. You have shutdown of all kinds of intrinsic cellular functions, and the cell dies. And so this is just to give you an idea of I guess, the nature of pertussis. As I mentioned, there are antibiotics, but by the time most people seek them, by the time they have that whooping cough, it does almost nothing to change the course of disease, although it will help block dissemination to new hosts. So people are treated with a mixture of antibiotics, and often they'll need some respiratory aid like extra oxygen support. They'll get an oxygen mask because they're just having such a hard time breathing. There is the DTaP or DPT vaccine, which is really ubiquitously used in the US and really effective. But just to give you a sense, if you look, here's pertussis here. This is looking at a lot of the diseases for which we have successful vaccinations in the clinic. If you look at pertussis and the number of yearly cases that were reported, and this isn't cases, this is actually deaths that were reported under the age of 5. There were 294. If you look at all deaths total, all of them were those children under the age of 5. So of all the people that died, they were all kids under the age of 5. And the reason that this is occurring is because it's so contagious and because it's still out there and circulating. A lot of kids will get infected before they've had the full sequence of vaccines and boosters. And so, because they have to be done at the right age spreading, and so if a kid catches it before they've had a full vaccination series, it can still quite easily cause fatal disease. So that gives you a sense of kind of how it's falling out in the US, where its niche is. Adults who get it, especially those who have been vaccinated, they'll get sick, but death is not a big outcome. All right, and so the last one we'll talk about today is Pseudomonas aeruginosa, and this one is a pretty interesting one that I think is one of the more successful pathogens in the environment, in part because it actually is everywhere, and it's what's known as an opportunistic pathogen. It only causes disease in limited circumstances, but it is absolutely everywhere and can grow everywhere. So it is really able to adapt and take advantage of its environment. It's a gram-negative rod-shaped bacteria. It can live in all kinds of conditions, soil, water, people, animals, you name it. It's really ubiquitous. And just to give you, and this part of the reason it can live everywhere is because it has very few absolute nutritional requirements. So it makes most of what it needs all on its own. And it has a very adaptable metabolic system where it can use energy sources or carbon or oxygen or nitrogen from all kinds of different sources in the environment. And so there are even stories of Pseudomonas growing in like mop buckets with dilute bleach. just to give you an idea of how robust this organism is. I mean, that's pretty impressive. So it's kind of cool. Its colonies have a really distinct fruity odor, and they're blue-green by nature. And so actually infected wounds, sometimes you can tell that they're infected with, I was thought about showing pictures, but they're pretty gross looking. But the wounds, especially if you have a skin infection where Pseudomonas is the causative agent of the infection, you get the wound. This is especially burn victims get these. Pseudomonas is really, really one of the dominant drivers in wound infection of burn victims. It becomes infected with this slimy mucoid biofilm that's produced by Pseudomonas aeruginosa, but it turns a blue-green color. And so that's a fairly good indication that it's Pseudomonas. And that's because Pseudomonas makes a pigment that actually is blue-green in nature. So in culture, it kind of has this cool blue-green color. If you put it on plates, it has it. You can see it here. But even in the live environment, it is sort of this blue-green color that you can see if you get enough of it there. And it's an obligate aerobe. So it won't grow in anoxic conditions like deep in the lung or deep in necrotic tissue or things like that. Okay, so pathogenesis. As I mentioned, it's an opportunistic infection or an opportunistic pathogen. It's everywhere. It especially takes advantage of immunocompromised individuals or people who have medical devices that are sort of constantly inserted. For example, indwelling catheters, things like that. It will colonize those tubes quite readily. Because it has such diverse metabolic and such diverse and minimal metabolic needs, it can cause infection in all kinds of tissues. So it's an example of an organism that doesn't just need to live in the lung or the skin. It can go everywhere. It is a pyogenic or pus-forming bacterium. So if you're infected, you will have pus produced at the site of infection. And as I mentioned, one of the ways to know you have it is the blue-green pus, which is pretty unique to this. So some common infections that are often associated with Pseudomonas. Lung infections, particularly those associated with cystic fibrosis. It is one of the biggest drivers of lung infection, cystic fibrosis patients, as well as in some other cases of pneumonia, but this is kind of the big group. Burn wounds are the second most prevalent place that Pseudomonas will take hold. So often it's a huge clinical concern in instances where the skin surface has been burned. It will often move in quite rapidly. As I mentioned, catheter sites, you can get a lot of Pseudomonas colonization and biofilm production there. It can cause ear infections and then eye infections, particularly those associated with contact lenses. So you can get biofilm formation, especially on contact lenses that aren't disposed of frequently, and it's a Pseudomonas biofilm, and you'll get a corneal infection. And it can be not nice. Okay, so how does Pseudomonas cause its damage? So it really has two major mechanisms of virulence. The first is by virtue of production of different pili, it's able to colonize a diverse set of different epithelial surfaces and even non-epithelial surfaces. And then as soon as it colonizes, it very rapidly starts producing this mucoid slime, which is like a biofilm that actually helps it become protected from host defenses, changes its metabolic function in a way it's not susceptible to treatment, renders it inaccessible to all kinds of things. So it is an example of an organism that uses that biofilm approach to really wall itself off and start replicating. It also produces two important, it produces more than these two, but these are two that we know a lot about and that have important functions. So these are simple AB type toxins. The first one is known as XOA, and the second one is known as XOS. XOA has that ADP ribosylation function and also targets E2F. So this is like what we talked about for diphtheria. It's shutting down protein synthesis, and that kills the cell. And it also, XOS is also an ADP ribosylase that targets G proteins. And as I mentioned, G proteins have diverse actions in the cell, and so this will also have very toxic effects on its targeting cells. Treatment is concerning. So it is associated with with a lot of clinical morbidity and some mortality because of the fact that it can dwell in so many environments. And the other thing is it extremely rapidly acquires resistance mechanisms. So a lot of the Pseudomonas that's sort of everywhere in hospital settings is already antibiotic resistance. So you have to take, when it does occur in patients, particularly immunocompromised patients, really aggressive antibiotic treatments, often simultaneously, and even then it can take over. It's kind of hard to get rid of once it takes over far enough. So the other element is just hygiene. Strict hygiene practices are absolutely essential because it's ubiquitous. And in hospitals or settings where there's a large population of immunocompromised individuals, it's one of those organisms you just have to be very, very aware of and take strong and rigorous measures to get rid of out of the environment. So this is one that in the clinic is a big deal for those patient cohorts. All right, so that is it for bacterial pathogenesis. Does anybody have any questions for the second half? No? All right, I think we're out of here early so you guys can enjoy the day.